Today we'll be talking about cells. As we know, all living organisms are composed of cells, and the development of these cells began with a research of a scientist named Robert Hooker in the 1700s. He observed the slices of cork and he made up this name called small units, I mean cells. This theory has three different principles. One of them is all organisms are composed of one or more cells. The second is the cells is the basic living unit of organism. And the three is all cells arise from pre-existing cells. Although cells vary in organization, size, and function, all share three structural features. As we know, all cells possess a plasma membrane, defining up the boundary of the living material. All cells contain a region of DNA, which stores genetic information, and all cells contain a cytoplasm, everything inside the plasma membrane that is not part of the DNA region. So far, we studied that there are two types of cells. One is the prokaryotic, which means before nucleus, and the other one is the eukaryotic, which means the presence of a true nucleus. All prokaryotic cells are typical bacteria, cyanobacteria, and archaea, and those protists, fungi, plants, and animae belongs to the eukaryotic cells. Well, since we have already classified the different type of cells, which are prokaryotic and eukaryotic, we are gonna talk about the different kingdoms. In this case, bacteria and archaea belongs to prokaryotic cells, and bacteria have a small single prokaryotic cell. The nucleus is not enclosed by the membrane, and some form change or mats. The bacteria cell has three different shapes. One of them is bacillus, which is this shape is cylindrical, and the very common is Clostridium botulinum. Another type of bacteria is coccus, symmetrically spherical, and you have two types of coccus, streptococcus and staphylococcus. The spirillum, there's another cell called spirillum, a spiral shape, and have one or more twists. Some of the main characteristics of a bacteria cell are that those are single cells organisms, those reproduce through cell division, those are also arranged as long chains and are called strepto. They have a thickened cell wall structure made of a carbohydrate peptide glycan. Now we'll talk about protist cells. Those are eukaryotics, and we usually find it in water. Some of the characteristics are well known for their motility. There are large single eukaryotic cells, which includes a nucleus enclosed by their membrane. In our experiments, we're gonna use a slim mold of a Fusarium foliiciforum, which belongs to the kingdom Protists. This unicellular organism contains all the metabolic machinery for independent existence. What we're gonna do today, we're gonna drop one drop of this wet mouth into a slide, then cover it with a cover slip and put it on the microscope and see if we can find some movements or some of the different motions that this cell can be recognized. As we know, movement of the organisms occurs as the solid state cytoplasm at the advancing tip pushes against the plasma membrane, causing the region to swell outward. The solid state cytoplasm flows into the region, converting to the gel state along the margins. Now we'll see fungi, which is one of the most important kingdoms. Um, all fungi, all cells, also walls contain polysaccharide diphthein, which plays an important role of this composer in the ecosystem. Those are also essentially for recycling nutrients. Today we'll work with a Saccharomyces cerevisiae, and we'll see some of these cells have different shapes. One can be flagella, cilia, cephalopus, and we also notice that in the fungi kingdom, cells can be unicellular or multicellular. In this case, those that are unicellular are called yeast, and the ones that are multicellular are called molds. Plants are also multicellular forms of cells which are specialized uh, in doing their own food. Uh, today we're working with Elodea leaf cells. Those young leaf are growing tip of Elodea are particularly well suited for studying cells structure because these leaves are not only a few cells layer thick. Um, as we see in the picture, we have an Elodea cell at 400 times. We are gonna observe that there's some green bodies on the cytoplasm. Those are the chloroplasts, which are organelles that function in photosynthesis and are typically found in green plants. We are also gonna locate the dark lines running parallel to a long axis of the leaf. Those are the air containing intercellular spaces. Um, we also have a video of it, and we'll see that this cell is a three-dimensional object. Therefore, we're able to see the cell vacuola, which can take up from 50% to 90% of the cell interior. Because the vacuola in LDA is transparent, it cannot be seen with a light microscope. The chloroplasts inside of the cell are surrounding the nucleus and occur in the cytoplasm, so they will appear to be in different locations depending on where are we gonna focus on in the cell. Another form of multicellular form of cells is animalia. Today we are going to use our own chick cells in order to see the different parts of the cell in the microscope. We're gonna use a toothpick and pull some scrap from inside of our chick. Gonna stir the scrubbing into a drop of distilled water and put it on a clean slide and add a cover slip. Because of the cells are almost transparent, gonna decrease the amount of light entering the objective lens to increase the contrast. We'll see the nucleus, a central located spherical body with the cytoplasm of each cell. Now, in order to stain our cheek cells, we're gonna use a solution called methylene blue, which will help to stain the nucleus darker than the surrounding cytoplasm. Um, we'll see the different parts of the cell, including the cytoplasm, the nucleus, and the location of the plasma membrane. Many of those cells will be wrinkled due to their thin, flexible nature. In overall, what we've learned during this lab is the different kind of cells, and we also took some pictures from it, and we'll see the different shapes and uh, how some of the particles circulate in it. Um, it's a really interesting topic, and I wish we could go in deep and see more cells and the different functions they have and the different shapes and everything else.